Good morning and welcome to the 2026 CRB webinar series. Today's webinar is going to discuss the ongoing work of the California CRaFT program. I am Caitlin Stanton with the Citrus Research Board, and I'm joined by Melinda Klein, who will be introducing our speaker today. We encourage you to ask questions during the webinar using the Q&A function, which can be found at the bottom of your screen. All questions will be answered following the presentation. With that, I'm going to pass it over to Melinda, who will introduce today's speaker.
Thank you, Caitlin. I'm pleased to welcome today's speaker, the Citrus Research Board's own integrated pest management entomologist, Dr. Ivan Milosavljevic. In his role at the CRB, Dr. Milosavljevic is responsible for developing applied citrus pest research projects with industry input. As part of that work, he is leading the California Citrus Research and Field Trials Program, also known as California CRaFT, which is focused on strategies for mitigating Asian citrus psyllid pressures in commercial groves in the state. Today, Dr. Milosavljevic will provide updates on ongoing program work, both in the desert and more recent work that's expanded into the Ventura County groves. Ivan, please go ahead and share your screen to begin your presentation.
Thank you, Melinda, for this wonderful introduction. Thank you, Caitlin, for organizing the meeting. And I'm happy to be here in just a 1 week. It will be 3 years now since I have joined the CRB. So I'm very happy that after 3 years, and really the CRaFT program has been the first one that I've really started working with since I joined the CRB. So I'm happy to share some updates from the program after these 3 years and what we have learned so far, and just to share some key updates and learnings from the program so far and what we have in plan for the future years moving forward. So the topics, this is a slide you can go, and this is the link to our webpage. We have our web pages updated with the new information on our CRaFT. If you want to learn more about the CRaFT program, the 2 different types of programs that we want to start now in Ventura with the new program, as well as the ongoing efforts in the desert. Please go ahead and just scan the code. I will have this code up and running at the very end of my last slide, but you can go on the CRB webpage and just go in the top right corner. I believe there is a CRaFT tab that you can just tap on and see the most current information. This webpage also has an application link for the Ventura growers. We're expanding into a Ventura in 2026 now in July. And if you want to apply for the program, you're interested, you will learn a little bit more today about that and what we plan to do in Ventura. But if you want to apply, and please do so on the webpage and contact me. There is a contact information to my email and my phone number. So please just don't hesitate to contact me at any time. The topics that I will be covering are quite broad. I'm quite sure that most of you are familiar with the ACP and HLP situation in California, but I really need to touch base on that before I start with why the CRaFT program was organized in the first place. Then I will share the updates and key findings. I will share the information on the dogs. We have found the dogs to be very effective in the desert and very helpful to our program. So I want to share the results on that. I also want to share the expansion into Ventura that we plan to start in July with the biocontrol releases and the self-release cages with the Tamarixia. And I will touch briefly the importance of the ant management, because that is of particular interest in California, as well as in the Southern California, where we have a lot of issues with the ants. They're not just a problem to ACP, they're a problem to mealybugs and scales. There are different types of ants. We have problems with the Argentine ants, but they're also a problem to ACP. So even though they're not like a direct management or mitigation being suggested for the ACP control, they are interfering with biocontrol. So the ant management is very essential. So with that, I'm just going to jump into a situation with the ACP and HLB. Just a brief overview, I guess. They're both invasive. So this is a pest complex that is invasive, and as with many other invasive pest complexes, it's very hard to control from the start, because they get into this new environment that is free of their natural enemies, free of management options. They get oftentimes undetected, in this situation and in many others, they would get into these new environments through a major hubs, like import hubs, like LA and San Diego in our case, and they will freely wander into, like, very slowly into these urban vast environments that have a lot of different plants for them to feed on, and slowly build up before they reach the commercial areas and before they become a huge problem to us, to our industries, right? So in a case of the Asian citrus psyllid, as it's called, the Diaphorion acitri, or ACP, it's a similar way. It was first detected in 2008 in the San Diego area. A few months later, it was already found in the LA area. And after 1 year, it really got established throughout. It was found everywhere in the Southern California. So it became a huge problem. Then, as in many other pest complexes with the disease and the vectors that transmit the bacterium, in this case, that transmits the disease of a plant or a human, we would see a lag effect before the disease comes or becomes evident. So that was the same in this case. In 2012, we found the first site where the CDFA did in Hacienda Heights in the LA area. And now we have an issue in these urban environments. So a little bit about going back to this pest vector complex. The ACP, I'm quite sure all of you know how to identify the ACP, right? It's a very distinguished, in our case, it's the only psyllid that we have in citrus. It has this like a 45-degree feeding posture, as you may, and it has this very distinct characters on their wings. The adults are, they can fly. They're not a good flyers, but they can fly. They can cover some miles, but they will lay eggs on this flush. And this is very important to us because the egg laying, as well as the nymph development, which are there immatures, it will be restricted to this new growth or a flush of a citrus, right? So they're quite less mobile. The eggs will develop in just a few days, a couple of days, like 2 to 4 days into these nymphs, as we call them, or immature stages. The nymphs are very similar in appearance. There are 5 different nymphal instars, and they will go through these nymphs in just 11 to 15 days before they become, again, the adults. And the adults can live for months. So they have a very fast developmental cycle. Oftentimes, many times, you will have all the life stages on one plant if you have an issue. So the management has to be coordinated in such a way that you have to manage both the adults and the nymphs. And the nymphs will feed on this flush, and they will feed on a phloem, right? So it's very rich in sugar. So they will poop these filaments, as you can see them on this bottom left or middle left bottom figure. They will have these excrements that we call the honeydew, and they will attract ants, for example. They will nurture them and protect them from natural enemies, but that's how they feed. They feed constantly until they become the adults. This is also very important because they're not a big problem to our plants, because they will not do a lot of damage to our plants on their own. But because they're a vector of a class, or I call it a class, or a CLAS, the Candidatus Liberibacter asiaticus, which is a causative agent. It's a bacterium, right? It's a causative agent of a deadly citrus disease, as we all know it, as a HLB, or a Wanglong being. And once they become a vectors, then they become a problem, because the disease, you can't manage it in such a way that you can control the disease, manage the disease. Once it's established in the plant, it's very hard to detect, and it will cause a lot of problems to the plant, and eventually it will die in just 5 to 10 years. It will still produce fruit in such a way that will be malformed, bitter juice. You will have a premature drop, but essentially it will kill the plant. So this is a problem, not a problem because they're a vector of this deadly bacterium that causes this deadly disease. And in this case, as in the case in many vectors, you need to reduce the number of vectors. So the management has to be focused on vectors, because if you reduce the number of psyllids, then you will reduce the number of disease incidents, and that's the only way right now you can manage this disease in this pest complex, and this is what the focus is on in California. The problem in Florida, for example, the pest complex, like I said, it's invasive. It comes from the Indian subcontinent and Southeast Asia. It has slowly moved throughout all the citrus growing in regions in in the world. It's present in Brazil. It got it into Florida very early, much earlier than here in California. It caused a lot of problems because they didn't manage it properly on time. And it with their mild climate and their efforts not being directed toward like right away towards the psyllids, it was very hard to stop it, and it killed almost 80% of, I believe the production was reduced by 80% since 2016, which it's a catastrophic effect, really. In our case, we were, I would say, more lucky, but also we got more prepared over time because what happened in Florida, and really our problem started in urban areas right away. And because we have so many different like citrus producing regions within the state and different climates, and the management efforts that we have put in these urban areas, we didn't give it a chance to establish properly until now. Like not until now, but even until now. So we are seeing these trends because, like I said, it was first introduced into the San Diego and LA areas, and it moves into an urban areas. It's an urban problem for us still, like major like it's our urban problem. So we were giving a chance to fight it off in these urban areas before it got established or before it moves into commercial orchards, which we are fortunate enough that we can detect, and we detected this problem early at the earliest stage, and we were able to manage it in these urban environments. So with the coordination from the industry, the CRB and the citrus industry in general, the CDFA, as well as the UCR, there has been a lot of different battlefronts in the urban areas, and I call it the IPM, the urban IBM. It might not be the IPM, but I call it the IPM because it has such a different components. It's very dynamic. We have had and we still have the biocontrol efforts that are being employed by the CDFA, the quarantine restrictions, the tree removals, the sprays after these detections of positive trees that have stopped this disease in the pest complex, and reduced the numbers to much lower numbers than what we see in other regions. And in combination with our climates in different citrus producing regions, like we have a desert citrus in the Imperial and Coachella valleys. We also have the coastal citrus and the lemons in Ventura, and we also have the intermediate in the Riverside area. But with all these different climates, we didn't give it a chance to properly establish. We do see this is the tally up to date. Like we have found the CDFA about 10,000 infected trees that have been removed. Sprays have been employed, as well as the psyllids have been collected, on time. The biocontrol program that I'm going to show in not just a few slides, but like back in my presentation, later in my presentation, it also contributed a lot to the reduction of these vectors because we were able to detect the psyllids early, and then the biocontrol program was started very early, and the wasps that have been used by the state, the Tamarixia radiata, has done a really good job in reducing these vector numbers. Like I said, the important part was to reduce the vector numbers before it moves into commercial areas, and the battle is still ongoing in these urban environments, and this is helping a program and commercial and the citrus industry in an enormous way that we are not seeing psyllids in our growth yet in big enough numbers to be established really and to cause any damage. But in commercial areas, we are prepared in some way, but we are not really we don't have a problem in such a way like in Florida, so we don't have a chance to develop a good program. We're not panicking, and we need a program early. So, but not a lot has been done in commercial growth to really stop the vector as it moves from these urban environments in such a low numbers into a commercial orchards. So the CRaFT program is really the first one that is dealing with this in California, and it is being funded by the USDA, by the governmental funds, and the HLB MAC group. So that stands for Wanglong being the HLB, and the multi-agency coordination or MAC group. This group was initiated in 2013, as you can see on the slide, and it really focuses on funding the shovel-ready research for the citrus industries. It does have members from the California, Florida, and Texas, as well as the USDA members. And it's a very important program that funds the industries in these 3 different states, as well as in other states where citrus is being reduced, but majorly in these 3 major citrus producing states, and whatever it's needed to be done in commercial grows and from the grower perspective, right? So the CRaFT program, being the Citrus Research and Field Trials Project, was really, there are 3 different CRaFT programs. There is a Florida one, the Texas one, and finally the California one that was the last one to be funded, our program. The Florida was really initiated in 2019. That was the first one, and each program focuses on different management perspective, on different based on different needs of these different regions, right? So the Florida is really focused on funding the research on resets, rootstock science, plants and soil nutrition, etc., etc. The Texas CRaFT project was started in 2021, and it started really. The focus is to study a citrus grow replants as well. Our program was the last to be funded. The funds started in 2021. We needed some time to establish the program, so the program started really in 2023. But it's the very one that focuses on the ACP management inside these grows. It's on early management, on low numbers of ACP, and it focuses on providing the best possible mitigations and monitoring tools for these pests at their early stage of development in our grows. So how the program functions, for people that are aware with the CRaFT, they already know how we function. The application process, there are application time windows that we have had in the past, and the application remains open on our webpage until we have a certain number of applicants applied for the program and satisfactory numbers and acreage in the program. The growers will apply on that web page with their acreage and the desired blocks that they want to see in the program, then we will coordinate with them. So they will select from these mitigations that are available on the UCIPM guidelines or the UCIPM web page that are being suggested to fight off or control the psyllids. So these have been developed from different regions and including some of the California data, but they're being suggested to growers as the best management practices. And these are only the additional mitigations. The growers are not being told to stop doing what they're doing in their grows. Whatever they're doing, have them in controls. And on top of that, these are the additional mitigations that we have provided to them to select. And then with our coordination, they would apply those mitigations, and then we would evaluate or assess how they do over time. The initial mitigations that we have had in our program we have divided them in preventative and threshold-based mitigations, as you can see. On the left side of this, left part of this slide, or left-hand part of this slide, you can see the preventative mitigations that are being applied once, maybe in a lifetime. And this part focuses on preventing psyllids coming into the grows, really, in such a way that you would plant either live plants around the growers or a barrier mesh fencing, as you can see, as well as on the trap crops that might be more attractive to them than a citrus. And then we had also the threshold-based mitigations, or we still have them, that are being based on the numbers. And I'll get into the numbers in the next slide, but it's really, those are the ones that you can see on the right. We have additional sprays, like insecticide sprays, additional kale and clay applications that is very important for the desert growers. We have the biocontrol releases. Lacewings have been the ones that are commercially available and mostly applied by the growers. And then we have the ant management that is very essential to control the psyllid as well. And once we have all the mitigations and acreage, we assess and we confirm the best practices. We compare against them or across them. And essentially, after a year being in the program, the growers are being compensated for whatever they have applied during that year of participation, or also for the participating acreage. So they get reimbursed, and then they continue with the new year into the next year, right? So what do we provide as part of this deal is that we provide the regular numbers or updates to them and what happens in their specific blocks. So most of the growers will have multiple blocks. They will have multiple different issues, multiple mitigations, right? So there is no limit to how many blocks you can apply with or mitigations that you can apply within those grows or blocks. we focus on our efforts on what has been suggested by the UCIPM methods, and that is being like sampling, visual sampling of flush inspections, tap sampling by doing the tap samples of branches. We also employ the traps. And also what we do have and what it's not up there is the dogs. We have found the dogs to be very productive in the desert systems. And I'll jump into that in just a few slides, but they have been employed by our program and they have been found to be very complementary to whatever we are doing in providing the growers numbers. The numbers, so we started sampling on both the outskirts of the grows and in the middle. We would use the 10 trees for each direction, cardinal direction, as well as in the middle of the grows. And we would do two flush samples as well as two tap samples, and the traps will be replenished every month as well. But we would provide these numbers like the visual and tap samples every 2 to 3 weeks, and then the trap numbers will be compiled about every month, and then the dogs will be. We started by providing them, and I'll jump into that quickly, but we started by providing them semi-annually, but now with the help from the Imperial County Pest Control Districts, citrus pest control districts, we can employ them even more frequently. So we're getting a much better numbers now. But all these numbers are being generated into the reports, and growers get them. As soon as we get them, we generate the reports and we send them out to participating growers, individual growers, and they have the clear picture of what is going on in their numbers, in their grows and blocks. So these are the results after the 3 years, being the 3 years in the program. And this is very interesting because the most, as you can tell, of our acreage and the most blocks are being located in the desert. So the program has been very functional and very representative in the desert. And again, with the help from the Imperial County Pest Control District, we were able to expand the program in 2025. So we started with about 1,400 acres, mostly being in the desert, and we had a few blocks, like 6 blocks in Ventura and Santa Barbara, about 55 acres. But then we added a lot more acreage in the desert because of their participation and interest in our program. And the program really functions in the desert, and I'm going to explain that with the next few slides. But we were not able to see any difference between the treatments. Some of the treatments that are being advised by the UCIPM were not available to the growers. So like trap crops, we couldn't start those. And those had to be jumped to control options, right? So the control blocks. But as you can tell in the right lower hand side of this graph, there is not a much difference really between the numbers. And that's majorly because the numbers were so low, as you can tell from the right upper side of this graph, that the numbers started a little bit higher in these areas, but then they immediately got down, and we really don't have enough psyllids to compare against the treatments. So whatever the growers have been doing so far in the program has helped a lot, and the numbers were low, so they were doing it the right way at the right times, and they were controlling the psyllids or reducing their numbers to a lower numbers, and essentially they didn't have enough issues. The additional mitigations did not show any effects so far. We do have them as an option, and the growers say that or the participants, they do help. But so far we couldn't tell the difference because the numbers were so low. The citrus varieties that we have, again, the representation was very biased towards the lemons. As you can see, for now, we have about 90 to 95% of being the lemons and grapefruits combined. And that is being fine because most of the lemons are being grown in Ventura, as we know, as well as in the desert, and the grapefruits add to that. But with this mitigation efforts, we haven't seen much of effect through our program so far. The monitoring, however, provided a very interesting data, and this is something that we really focused our program on moving forward, because the monitoring, we all knew this, what you see on this slide. We all knew that the ACP would be a perimeter problem, right? But we didn't have enough data to show this, and the data hasn't been analyzed so far to show that this is a perimeter or outskirt problem to start with. And this is striking to me because they're so even. Like I said, we had the samples on east, west, south, and north portion of the groves, as well as from the center. And all of these data is being based on the visual inspections of the flush and the tap samples combined. But it's very even across like the east, west, south, and north, as you can tell. Like all of our samples, the majority, it's very evenly. All of the psyllids that have been found, the proportions that are being found on east, west, south, and north, they're not very different, even though I expected them to be different because different groves have different environments around them. They have their neighbors, different like um, uh, different visibility to these psyllids, so or they have major roads on some ends, but we haven't seen the effect, any edge effect, except for the major edge effect compared to the center. And on the right side of this graph is the center, and we had only a few samples being found in the centers, and those were found in the very young age growths that are very short, or in the growths that are on in the bottom from the hill. So they will go down if there is an urban like a tree, they can fly down to that grow in the middle and establish. But in these major blocks, commercial blocks that are flat, all of them will be initial detections will be on the outskirts. And this is very important data, and this is based on our field tech data, right? The visual inspections and like I said, tap samples as well. But they're very difficult to detect, especially if you are focusing on just the 10 trees per side. They can cover more. This is a standardized way of how you sample these blocks, but they can go undetected as well, especially in such a low numbers. So that's why I want to bring up the dogs, and this is why I mentioned them, why they're so helpful, because they can cover more area. They go with the handler. They sample the perimeter of the blocks, and they just walk the grow, and they alert wherever they smell the psyllid. They work on the scent of a psyllid, right? And they're very helpful in such a way that you can cover a lot more acreage around these areas. They can confirm the finds by the handlers, or they can find something that handlers haven't been found. So they are really an additional monitoring tool, very helpful to the desert grows, especially because we had such a good representation and such a small numbers. It gave us enough space to evaluate these dogs to to have a better data from them. We started, like I said, with the semi-annual inspections with just a subsample of crows that we can sample. We were limited to that because of different constraints. But in here, and I'm going to point in the right, if you can tell, the right portion of the slide, you have surveys by period. You have the blocks that have been sampled and the time frames when they have been sampled. But overall, we have found so the way they function, they would go around the blocks with a handler. They will stop when they smell something. And essentially, the handler will confirm whether there is a psyllid on that tree or not, where they stopped, or they behave differently. And this is very important because there is a detection, right? And there is an alert first by the dog, and then the confirmation, and that goes into a detection rate. So even the dogs can miss, or the handler will not find. Even the dogs can smell it. The handler might not find it on the tree because it might be upper in the tree. It might be somewhere place else. It's very hard to inspect the whole tree, especially when there is a lot of flush on it. But the detection rate will be the proportion of alerts versus the confirmation by the handler. And the detection rates have been very high. And this is very surprising, like 88% in this first run over like 2023 to 2025. As you can tell, they have been very constant, and they have found a lot of eggs and adults. As you can tell on the right side in the data, we have found about 154 adults, 1,000 nymphs, and 1,000 eggs during this period, which is a lot. The striking is that in 7 blocks out of 70 that humans haven't found them before that. So how we would compare them or run them against the humans in our scouts. We would have the scouts first sample these blocks in our way, like a standardized sampling process of 10 trees on the outskirts of each cardinal direction. Then we would have the dogs in 3 to 5, maybe a week after that, going into those same grows, same areas, and sample the whole perimeter. And in 7 out of 70 of human, as we call them, human negative blocks, we have found, that's 10% of blocks we have found the psyllids. And this is very important because if the human missed, the dogs have complemented that, and this is very important because we're dealing with vectors of a disease. So we need to find the psyllids early so that growers will know if they're present in their blocks or not. So this is a very complementary and very good way of showing or supporting the field text or whatever is being proposed by a human scouts to be done inside these grows to monitor the ACP. And even striking is the next phase that we have done in 2026. So this year, early this year, and we have sampled all the blocks with the help again from the ICC-PCD that has acquired some of these dogs. We have sampled all of our blocks in a very short time window, so in just a few months. And we covered both the desert and the coastal areas. And again, the confirmation rate, the detection accuracy has been very, very high, about 90% on average. We did see a better effect in the desert because in coastal areas, you really can't focus on these dogs. If there are more trees infected, there's more smell, more scent, they will alert in more ways. They won't be as productive as you need them as compared to the desert, for example, where you only have like a few psyllids or maybe one psyllid being sitting on that tree and establishing a population maybe, but you need them to go around and find that ACP for you that might be difficult by the handlers, right? Or not the handlers, by the scouts, the human scouts. The very striking is that out of the 190 desert blocks that didn't have a human detections, the dogs confirmed in such a short window in 41 blocks psyllids. So in 41 blocks, the humans have missed on ACP, and that's like a 21%, as you can tell, 21.6%. In 21.6, that's almost 1/4 of the blocks that we have. The dogs have been finding these psyllids. This is very important because this tells us that maybe handlers or the scouts will miss, but then you have the dogs that will go behind them and find the psyllids. And this is important because you need to establish your management options and management program right away when the psyllids are establishing in these grows. And if you have a problem that it's in such a low numbers, the dogs have been found to be very effective. It is very like the ICC-PCD has the 2 dogs right now, but very noteworthy is that I need to mention is that the initial program with these dogs and the services, they had about 7 dogs. They have run this initial study where they have found that very important is reproducibility, because you will not have only one dog, you will have multiple dogs running all of these blocks. So, but all of the 7 dogs that they have had initially, the way they trained them is that they would have a vial with the ACP, they would put it on the tree, and they would run the dogs, and each of these vials, it will be a different vial for a different dog and different in the same setting of trees. The detection accuracy was very similar across all these dogs. So if you train them properly, you will have a very efficient dog, and it was above 90%, which is very important. This is exactly what they do. For example, with the detector dogs at the airports or maybe with the US Postal Service, they would have that like a standardized confined environment where they would train them and see how accurate they are, or what is their detection rate and how many dogs you can have the same detection rate across. And this is exactly what is being done with these dogs off limits, off site, when they're being trained to be employed into these grows. Okay, so this is just a few slides on how we provide the data or get the data. We would get them from the services in such a way that we would have a block where they have sampled the detection where it occurred. You can see it in the middle, like a PDF file. The green arrow, this is a desert block that had only one detection in the left corner of this block. And then all the data will be provided to these growers, and essentially the photo will be made by a handler just to confirm that there was an ACP in that grow and there was a productive alert. And you can tell that from the right hand bottom photo that it was really the adult that was sitting on that flush. Like I said, in Ventura, it might be a little bit different because you would have too many trees to sample, but still there will be a lot of different sittings and confirmations. And it might not be as productive as in the desert areas where you need that immediate detection right away in low numbers. But yet again, the detection rate was quite high, to my surprise, because I was very skeptical because the flush and the ACP levels could confound where the dogs will alert or sit, but it was very high still in the 90s. But this is like one of the trees. So this PDF file, for example, that we get will be quite longer because each of these detections will require a photo and confirmation by the handler. So I just pulled up one of them on the right-hand side of the graph or the slide. You can see just the first detection that has been made, just one green arrow in that block, and then the photo from that particular tree. Okay, so that is what I have so far. So what we have learned is that we can really rely on our monitoring tools. We can we have found a way to see where they're most prudent and where we can apply them, as well as the dogs as being found as something novel or a novel tool that we have been developing through our CRaFT program. The new program is really interesting because we have had so much success in the desert areas and so much participation. In Ventura, not so much, right? Or the Santa Barbara. And this has been bugging me for years. So after talk with so many growers from Ventura and attending the meetings, they have expressed the need to really for a different type of a program. So they wanted to see more of biocontrol in their grows. And this is exactly what we are providing to them. And like I said, we're starting in July with our new program. And what we really want to see is the biocontrol efforts being from the urban environments being implemented or transferred to these groves. And with the help of Dr. Raju Pandey, who is also an entomologist at the CRB, these self-release cages were developed, and we're trying and still being evaluated, but we're trying to implement this into our CRaFT program in Ventura and with the Tamarixia wasp that I'm going to explain in the next few slides. And I'm going to jump back to the cages, but we want to transfer something that has been working from the urban environments and it was very effective into a commercial grows because it's not being available to growers commercially. It's very hard to reroute and establish. So we are providing a new tool to these growers in Ventura, and with the low input IPM that they have programs, as well as organic grows and ranchettes, this might be a perfect environment for this program to get established and lower the numbers where we see them in higher numbers compared to the desert citrus, for example. But the Tamarixia problem, or the Tamarixia program, sorry, it started really when we first had a detection of ACP in urban environments. So it's also a wasp, not also, but it's a wasp that was introduced from the same native region from where the ACP is, from Pakistan, which is a part of a native range of the ACP. It's a ectoparasitoid. It's a very specific natural enemy of the ACP. It attacks the ACP nymphs, the later stages of 4th and 5th instars, so the bigger nymphs. And it's like in an alien movie, and you will see that in just a few slides, but it will, the adult wasp, it doesn't affect the humans. It's only very specific to psyllids. It will lay an egg on the bottom of the nymph, and then the larva will eat out the nymph from inside out and eventually emerge as a new wasp. So this is how they reproduce. They have a very fast reproductive cycle, and they have been very effective in urban environments. So there has been a lot of steps that you see here on the right side of the right-hand side of the slide. A lot of steps need to be covered before these wasps could be released in our urban environments. There was a risk that was assessed in the quarantine facilities, whether they're very specific to our nymphs or the ACP, or they will attack something that we have, some of our native fauna, for example. So they, after all these assessments that really speed up from 2010, 2011, it was done at UCR. The wasp was found to be very effective and very specific to nymphs. And it was finally released in 2011. And to date, about 35 million of these wasps have been released in backyard citrus. And more importantly, that I'm going to share in what we are trying to do with our cages, there have been different lines. We call them the ISO cage lines that were established. There are just different cages in this quarantine, and they represent different regions from where these wasps have been collected in Pakistan and brought to the quarantine facility. So we kept the original lines, 17 of them. And this is very important because if you have a one cage and you put them all in one cage, essentially in the quarantine, in these cages, they will lose an effect because they won't be as prudent or as potent, as if you mix them together. There won't be enough genetic variability within these wasps. They won't be they will be more laggish, and they wouldn't be able to attack the ACP as we want them to be. If you have all these lines and you combine them when the time is for release into a single cages, and then you mix up the genetic material, then you will have a much better parasitoids or natural enemies to release and starting pool. And it's just essentially you would release them on the trees and you will assess what they do. And this is exactly what we have done. And I was a part of this program with the CDFA. This is the data from 2015 through 2018. And we have seen a very high parasitism level across these different regions in urban environments. All of these sites, we had 30 different urban locations where Tamarixia hasn't been released, but in those areas it was released. So we were trying to see whether it got established, whether it's doing a good job. And it really, the blue arrows that you see is the trend of ACP numbers over time over those 4 years. The purple lines, the purple curves, is really the parasitism rate that really followed any peak of the white curves, which are the ACP numbers. Whenever the ACP would peak, the parasitism will follow. This is called a lag effect. And after 4 years, the 75 to 90%, depending on the different life stage of these psyllids, it got reduced. So the program really worked. It was a massive program. The psyllids were released across all these areas, and they were evaluated, the parasitoids, and they worked. They really helped the program. It might be that the climate added, whatever the CDFA is doing with the tree removals and these quarantine restrictions of movements of live plants, but also biocontrol contributed to this overall cause. And they're very effective. And this is exactly what they do, what I explained in an Alien movie. I just have a few moments to share. But if you can look at the right-hand side of the photo of the slide, the video shows how this wasp actually emerges from the adult nymph. And it's not just the parasitism, really, because the parasitism you can quantify by looking at these exit holes, but they're also doing a very good job through a host feeding. And what that means is that they will kill maybe every 5th or 10th. Nobody has done this in the out environments, but it has been found that a host feeding, they need to probe the psyllids as they find the suitable host. And as they probe, they will mutilate or they will kill off a good chunk of these nymphs just by trying to find the suitable host to lay an egg. But you can't quantify that in the field. It's very hard. So what you can do is only quantify the parasitism rate. So these parasitism and these reductions might, the overall benefit of this program might be even higher with the host feeding. So they're providing a dual job for us, really. And this is exactly what we are trying to implement through our new CRaFT program, and to use these self-release cages for these wasps. This is something that we are still developing and we hope to implement in our CRaFT program on a larger scale. We have found them to be very effective in a few blocks that we have in Ventura and in Riverside. It has all the benefits to the growers that they can acquire from the commercial production systems. The wasps have been very costly to produce. The private entities don't have the access to these wasps. It's not productive for them. They're very costly, so it's not productive to them. It's not productive to the growers to buy. And growers are lacking really a good tool in the growers to be used. They're lacking this very effective natural agent that will fly away and find the nymphs and do a very good job in controlling the ACP. So the self-release cages were developed in such a way that you only have to maintain the plants and psyllid levels inside, and you only need a few initial releases of Tamarixia, so you don't need like a lot of these wasps to be purchased. And with just a few initial investment, you can have about 2,000 to 10,000 of these wasps being produced inside these cages in very short time window and being released at the time when you need them in your grows, and continuously as well, because you will use the curly leaf plant. This is what we use. It is a very good host for ACP, not for HLB. The cages will have, and I'll show that in next slide, different levels of these screens. You will have a very fine screen on all the sides of the cage, but on the top there will be a more coarse mesh that will allow Tamarixia to be released, but not the ACP. So Tamarixia is a much smaller than the ACP, and that mesh is perfect enough just to release Tamarixia, and it will go to the nearby trees, and essentially as it gets established into a nearby growth. And it's really working so far, and we hope to implement this on a larger scale in Ventura. Like I said, our application process is available online. I'll provide a code in a few slides, but please, if you want to join the program, if you want to see this is a perfect program for the, like I said, low input IPM grows, ranchettes, and organic grows where there are limited options for growers to use or choose from. So this is again how the cages look like, and in the middle photo you can see this more coarse mesh that allows Tamarixia to get outside, but not the ACP. And with that, I'm just going to jump quickly to ant management. This is something that we need inside these cages. This is something that we need in urban environments, and this is something that we need in our grows. And especially if you are in Southern California, where the CRaFT program is being implemented, or you have issues with the ACP, where it's as we call it, endemic, or it established over time. Ants have been a very, very big impediment to our biocontrol programs. And these are called the Argentine ants, Linepithema humile. And you can have millions of these ants just marching on a single tree. They will go for this, like I mentioned, sugary poop or a honeydew. And in return, they would protect all of these. Like, it's not just the ACP problem, it's also a mealybrug problem, a scale problem, but they're there for ACP as well, and really impeding all the programs, the biocontrol programs and the IPO programs that growers are trying to establish, as well as in urban environments where we are releasing Tamarixia. We have had a lot of issues with these ants because they are an urban problem, and they are also an industry problem, or they're an ag problem as well. And how they work, this is Tamarixia. This is just a slide from what happened in our urban releases with Tamarixia. They would just, when Tamarixia, it needs some time to find a host, like I mentioned. In the process, these ants will be all over these nymphs, protecting them in exchange for the sugar. They are liquid sugar feeders. And if Tamarixia approaches what they consider their like stock or nursery stock, right? They would just remove them or kill them off or bring them back to their nests. And like I said, there are millions of them. They have multiple queens, multiple colonies just below a single tree, millions of them. They go up and down, they collect the food, they bring down the food, they feed the brood or their young ones, they feed the queen, and that's how they function. So you need really to control them. You need to control these numbers. You need to stop them going into these trees inside the cages so that they don't disrupt all these wasps to being mass produced inside the cages. In the urban environments where Tamarixia is really establishing and being still released in high enough numbers, like I said, it's very costly, so you don't need to lose them in this way. And also in commercial citrus. So the next few slides will just show you how important this is. This is a study that we have done in 6 commercial grows with the liquid management of these ants. And I'm not gonna cover all the details, but we have tried to see if you manage these ants every month with these liquid baits that contain a very small amount of poison, they will essentially kill the workers and the queens, because they will get into their mouth whatever it's in the liquid poison and bring it back to their queens, to their nests. And essentially the colony will collapse once the queens are fed with the poison. And after a year and a half of this program in different citrus orchards, we have found that all of the natural enemies increased for the ACP, the mealybugs, the scales, including the red scale, that it's just being on the trees but not producing the honeydew, but still being protected by these ants. The ant control contributed a lot to this cause and maximizes the biocontrol efforts that we have had in those groves. The same thing happened with our new program and another HLB MEC funded project that funded the development of these different type of treatment options for these ants, including the gels that I'm quite confident that you all are familiar with the gels. But this time we didn't have high enough ACP levels, so we focused on mealybugs, for example, as a proxy species, and we have found the same effect. Once you reduce the ant numbers, you will have a much higher rate of predation and a much lower number of mealybugs. So it's consistent across all these sap-sucking pests that these ants are protecting from the biocontrol programs that we're trying to implement. And really, this is just a slide that summarizes this. If you want an effective biocontrol program, and especially for Ventura, if you want effective control of ACP in your grove, in your backyard, you will need to control these ants, because they will most likely, if you have them present, not allow these natural enemies to establish. And that goes not just for Tamarixia, but for the lacewings, whatever you're trying to release in these groves, it will have a much lesser effect. You will have a much higher cost if you don't control for these ants. And I'm going to end here. We have time for questions, but I'm going to end here with, again, the QR code that you can scan. And please go to our webpage, learn more. This is the slide that has really my information. It's my email is here. It's also on the webpage, ivan@citrusresearch.org. And I will take any questions if you have them for me. So thank you.
Thank you, Ivan. We will now go ahead and ask the questions in the chat. I do see there's one question, Melinda, if you want to start it off.
Sure. So it looks like there's a question about looking through your results in the blocks where the scouts missed. Was there a specific life stage that was found, or was there a stage that was found more often?
Yes, that's a great question. And I think the second slide that I have really gets into the weeds of that. If you look at the deserts, these are the blocks that have been missed. So we have found the scouts have missed 53 adults, 335 nymphs, and almost 700 eggs. So this is how much they miss. So we have found all the life stages in those blocks, not just the adults or something that it's easily being missed if they fly away.
Great. It looks like we have another question. Are the ant bait gels registered for use in California for commercial groves?
No, they're not. I'm still very optimistic, and I really want to see the gels being registered. David Haviland is leading this registration process. There is always something new from the DPR that comes up or the EPA that we have to deal with, and I'm helping from the industry standpoint to collect as many data as we can on the efficacy, on the off-target effects. All of those data have been compiled and submitted to the DPR, and it's really on David Haviland and their team that is leading the efforts and negotiations with the DPR, if I can call them that way. But I will invite you to maybe listen to his talk if you're from California. He will present in October in our a citrus meeting. So if you have time, please stop by and listen to what he has to say, and maybe you can ask the questions over there, or hopefully I will have a much better answer. I hope to have a better answer in the near future, but for now they're not registered. We can't use them, unfortunately, because they're very effective. I already, I can assume that all of you know that, but they're a tool that we need. So we need the end control, like I mentioned in these slides, so that we can control ACP, mealybugs, everything to a better extent. So I hope, and I keep my fingers crossed, and I'm going to do everything I can to help David and the team.
Next question is whether or not CRB has plans to extend the CRaFT program to Kern County, especially when dogs are so effective. The speaker asks if it could be that we're not aggressive enough with sampling efforts in the San Joaquin Valley, especially Kern County.
For now, we are focusing all of our efforts with the program on where the program is needed. And I think for now, we have the most problems in our Southern California. The numbers and detections are still much higher than what we see in the Kern County and up in the San Joaquin Valley. The focus for now is in the Southern California. I don't know how the program will develop over time. I hope we don't get there to really need a program in the San Joaquin Valley. About the sampling efforts regarding to that, I really don't have an answer to that. I know that the APCD and the Alliance of Pest Control Districts and the CDFA, they're doing a good effort in sampling in those areas as well. And they're finding, we have our grower liaisons as well. They're helping with the cause. Judy is over there. So she's really on point with providing the data, much needed data to growers. So for now, I don't see the CRaFT extending there because we have so much issues in the SoCal that we need to address at the moment. And I think reducing the numbers here will help having less and less detections being found in the Kern County as well. So I would focus the program for now here.
And I think the other point of the CRaFT program is that what we learn from these other areas will then be applied to other regions as they're needed in the state.
Yes, of course. Thank you. Yes.
And then I think we've got one last question is, considering the timeline for a hydrogel bait, the status to getting that approved is up in the air. What other ant control methods would you recommend?
I can't recommend anything. I don't think there is anything on UCIPM webpage. I didn't check last. I know that growers are using different sprays on trunks for different purposes, maybe, but they're also seeing some effects for a month or so. I don't think any of the sprays will help because the sprays will kill the workers, but you won't kill the queens. So whatever you are doing with the sprays, it won't affect their populations below ground. So you will have almost instantly the new ants coming out very soon. So for now, the focus should be on the way of how to control the queens as well. I don't have an answer to that. I've spent too many years working on these gels so far. I want to see them registered. So any type of bait, maybe the bait stations will be very similar to that. That will be my only option because you need a bait to be taken to the queens. I know that there is some work being done in urban areas with some like a pheromone strings that might deter them from the trees. That might be a option for some like a cages or urban environments where you have just a few trees, but not for the growers. Not for now, at least. They have to be evaluated on that level as well. But if the gels don't get registered on time, I'm sure that we have to go back and start looking at something because the DPR has spent a lot of money. I have spent a lot of efforts. UCR lab that I worked in, Mark Hoddle, David Haviland, all of us have spent an enormous amount of time so far looking into these alternatives to chlorpyrifos. And this is the one that it's very effective, it's sustainable. So I'm still giving it a high hopes that this will get registered. And like Melinda said, CRB is doing everything we can to help the cause. But I don't have any recommendation outside of that for now.
Okay.
Maybe the
That
tanglefoot.
sounds fantastic. I think
Oh,
we're at time, Ivan, so I think we'll leave
okay.
it there for now. But thank you again so much for your presentation today.
Okay.
I believe we
Thanks.
may be around for any kind of last minute questions. But otherwise, want to thank everyone for their time today. And webinar will be available to rewatch on the website, citrusresearch.org, in the next several weeks. To stay up to date with upcoming CRB events, please be sure to visit our website. Like I said, we'll stay on for a few minutes in case there's any other questions. But otherwise, thank you for your time this morning.
Thank you. Thank you, everyone.